So our next guest, you probably all have already heard of. He's very famous and a good friend. And Dr. Lee Hood. So Lee, come on out, please, would you? And Lee is, as you can see his titles currently. They change about every 2 weeks. He keeps inventing more things. The thing you probably don't know about Lee, or you might not know, is that Lee invented the very first DNA sequencer. And for those of you who are interested in this stuff, you do know that already. But what even you might not know is that actually there were 6 machines that were required to do that work of doing DNA sequencing, and he invented all 6. Glad to have you with us. So Lee, you've been busy as usual, and I've had the benefit at FIRE of hearing you a couple times talk about your work. Lee is now, I would say, the world expert, or one of the top, how many? 2? 1? The? Okay, the world expert in systems biology and in using computational understandings to further accelerate preventive medicine and predictive medicine and personal medicine. Is that a fair statement?
Yes.
Okay.
Wow. Yeah.
And Lee has created a thing, which you see right here, called P4, which he's been working on for a long time. And I'd like to, if you don't mind, tell us a little bit about that.
Sure.
Okay.
I will say that this is probably the most exciting time in my career of more than 50 years or so. And it's exciting because, one, we've clearly defined 21st century medicine, which is very different from what most people practice. And 2, it's really going to transform healthcare in many, many different ways. But let me talk about this in the context of 21st century medicine. I call it P4, predictive, preventive, personalized, and participatory, and the requirements for big data and so forth. I had the pleasure of participating in 7 paradigm changes that dealt over the years with biological complexity, but they framed what this 21st century medicine is really about. The first was bringing engineering to biology, developing these 6 instruments. And what that did is, for the first time, you had the ability to make high throughput measurements in biology. And these in time accumulated to the point where we saw the initiation of big data and the appropriate analytics. One of these instruments, the automated DNA sequencer, led to making it possible to do the Human Genome Project. What the Genome Project did for us was to let us view the enormous diversity present in each of us and correlate it with our either wellness phenotypes or disease phenotypes. The automated sequencer also required the joining together of computer science and engineering and chemistry and molecular biology, and that led to the conceptualization of cross-disciplinary biology, where leading-edge biology could drive a relevant technology, and that in turn the appropriate analytic techniques. I tried to persuade Caltech, where I was at the time, to do this, but the biologists were against it. Bill Gates made it possible to go to the University of Washington and set up a department of molecular biotechnology, the first cross-disciplinary department. And that department was unbelievably successful in pioneering tools in genomics and proteomics, in cell biology, and in computational biology. In the end, I decided the university was not the place to create an institute for systems biology for a variety of reasons, and I resigned in 2000, started that institute, the first Institute of Systems Biology. And of course, systems biology is really about realizing biology as an information science and taking a view of data that is holistic, global, integrative, and hierarchical. And we applied these tools both to biology and even in the very early days, the early 2000s, to medicine. And that led to the conceptualization of 2 formulations of 21st century medicine. a systems approach to disease we called systems medicine. And that very much involved technology and computational tools. And we came to the realization, as I said earlier, that healthcare should be predictive, preventive, personalized, and participatory. And of course, that vision leads you to the conclusion that there are really two major domains in healthcare. One at the time was virtually entirely ignored. Wellness on the one hand, disease on the other hand. So it was in 2014 that we actually persuaded 108 of my friends to undergo more or less a year-long project and to use what we called personal dense dynamic data clouds to generate information that let us optimize wellness and avoid and/or ameliorate disease. And I'll say just a little bit more about that in just a moment. And finally, in 2016, Rod Hewick, the chairman of Providence St. Joseph's, came to me and said, we'd love to have ISB become our research arm and you are chief science officer. And I saw this as a wonderful way to bring P4 medicine to the healthcare system. We've certainly done that. So the convergence then of systems biology, of systems medicine, of digital health, of big data and its analytics, and even social networks, all led us to articulate just much more sharply what P4 medicine is. And first of all, the technologies that we discovered ranged across genomic, proteomic, single-cell analysis, and so forth. And the strategies, systems-driven strategies, where you take a technology, one or more, and put it underneath a computational platform so you can make very, very high throughput measurements. And we'll talk about one of these in just a moment. On the computational side, again, the view of data is it should be absolutely global, that machine learning, that artificial intelligence, the ability to see patterns, what we're talking about here, is really key. And the ability to handle and integrate and model information that's hierarchical in nature is really critical. The data clouds that I talked about earlier are enormously diverse, and they allow one to capture both genetic information and environmental information, which when integrated lets you optimize wellness and/or avoid disease. And you can apply these data clouds to wellness and/or to disease. This is the essence of what precision medicine should be. As to the hierarchical nature of biological information, the networks that govern and regulate development, physiology, aging, and if perturbed can cause disease, are absolutely critical to understanding disease. They operate at many different levels. We can now capture information uniquely from all of those different levels. And one of the questions is how you integrate this kind of information together. So P4 medicine differs strikingly from contemporary medicine. It's proactive, it focuses on the individual, it's about wellness as well as disease. It uses the data clouds to integrate your environmental and genetic contributions. It looks at clinical trials as an absolute failed disaster as they exist. 20,000 patients, you give them a cancer drug, half of them, you give the other half a placebo, you capture the data. But the fundamental assumption in that test is that all the units are identical, and humans aren't identical, either genetically or environmentally. And what P4 medicine does is actually, it would have 20,000 individuals, each with their unique data cloud, and stratify them according to properties you're interested in. One property, for example, could be, can we create a biomarker that distinguishes responders from non-responders in particular clinical trials? The social networks are advocacy, recruitment, education, a variety of other kind of things. Here are the top 20 selling drugs in the US today. The Orange individual is the responder, the blue the non-responder. The best case is 1 in 4, the worst case is 1 in 25. With classic clinical trials, you have no idea how to distinguish responder from non-responder. If we had the individual data clouds, we'd be able to ascertain that almost immediately. So the vision for clinical trials in the future is they'll be two-stage. You'll get the biomarkers for the responders, and your second stage trial will be all responders, and even with as few as 50 individuals, if you have a 96% rate, the FDA will agree to do it, to approve the drug, as it did with Herceptin and Genentech some years ago. Two major and central themes, as I've said before, are wellness and disease. I think what's really going to be interesting is over the next 10 to 15 years, wellness is going to emerge as a major focus and determinant of healthcare. And I think, frankly, the market cap of the newly emerging wellness industry in that time will far exceed that of the disease industry. And how pharma adjusts to that, I think, is absolutely a fascinating question. The wellness project that I talked about earlier, the 108 individuals that together with Nason and Price, we analyzed these dense dynamic data clouds, as I talked about earlier. Complete genome analysis, we took blood samples every 3 months to look at metabolites, clinical chemistries, and proteins. We did the gut microbiome every 3 months in the first study, and we're able to get the ratios of species that were present. And we did digital analysis with Fitbit and other devices to give you self-measurements and so forth. Again, the data, when integrated, led to a library that we'd already created from the literature of actionable possibilities, which, if acted upon, could improve health and or let you avoid disease. These actionable possibilities were delivered to each individual by wellness coaches that not only knew psychology, but the biology of the wellness determinants as well, and were able and persuasive. 70% of the wellness suggestions were actually approved because of the coaches. I think this is going to be a complete new sector in healthcare, wellness coaches in the future. We created in mid-2015 a company because the other study had ended. Most of the pioneers wanted to go on. Airvale now has about 4,000 individuals across the states now. They're in all states but 3. And ISB has an agreement with Airvale to analyze the data. And in fact, that's what I'll talk about now. I hearken these data clouds as being equivalent to the Hubble telescope, which of course could look at the universe with unparalleled resolution. So too, these data clouds look at biology and at disease with a resolution we've never, ever had before. So we made 3 interesting observations. One, we could do incredible statistical correlations, taking the 6 major data types we had and looking at each individual data type and looking at data bits from each of the other 5 types to see if there were either positive or negative correlations. There were 3,500 static correlations, about 3,000 dynamic correlations. The 2 were quite distinct from one another. And this led to brand new insights, both into biomarkers, but as well into drug targets. And this is a completely new approach for pharma to approach drug target biomarker discovery. Number 2, we made the absolute fascinating observation. If we divided the individuals, the clients, into decades of 10 years, 20 to 30, 30 to 40, up to 80 to 90, we could see for each of the major blood analytes, clinical chemistries, proteins, metabolites, an enormously increasing divergence in the ability of people to control the analytes. We could use this to begin determining biological age as opposed to chronological age. Mine, for example, I'm more than 15 years younger than my chronologic age. And the ratio of biologic to chronologic age is a metric for your wellness. And it's a metric by which you can do fascinating N of 1 experiments, which I'm doing, to increase the divergence between those two numbers. And the final thing I'll talk about are state transitions from wellness to disease. And this is an example of a 57-year-old woman in January of 2017 who was diagnosed with stage 4 pancreatic cancer. We had 4 previous blood draws prior to the diagnosis. And for each of those blood draws, we took the 1,200 or so analytes we had, and we analyzed them one at a time for the 2,000 people we had at that time to ask, in this woman, were there any outliers from the normal statistical pattern we saw? And in fact, we beautifully saw a protein outlier way beyond anything any of the other 2,000 or so patients had. A protein that's in a Notch receptor pathway, a cancer pathway, a protein specifically expressed in the pancreas, and it's an ideal candidate for a biomarker and or for a drug target. But we went one step further. We developed computational techniques that let us study 9 different biological networks and determine whether or not they were disease perturbed. So in the first blood draw, no networks were perturbed. In the second, a single network was perturbed. In the third, 2 networks. And then in the fourth, 5 networks. So you could see an expansion of the number of disease-perturbed networks as the disease progressed. And what this means is really interesting. So what we can do in looking at wellness to disease transitions is create biomarkers for the very earliest stage of that transition. And it'll be a long period before you see any clinical events whatsoever. That early stage then means that is the point at which we should create a therapy to reverse the disease before it ever gets into the expanded disease-perturbed networks and irreversibility. And we're arguing that we can do this for every major chronic disease. The one we're starting with is Alzheimer's, and we're already quite far along. And we can see in a 3 to 5 year period the possibility of identifying the earliest stage of Alzheimer's years before what we can do now, and B, the ability to reverse it back to normal. And we think within a 5 year period, we'll be able to reverse more than 80% of the Alzheimer's individuals. The cost of Alzheimer's for the 5.6 million people that exist today is about $500 billion. And imagine if you can eliminate more than 80% of that in the next 5 years or so. So this is my last slide, and it again gets back to the data and the importance of, from data, creating correlations and or patterns, and from those patterns creating hypotheses, and then doing perturbations. And I'll say more and more N of 1 perturbations in humans are really going to be key in understanding disease complexity and in analyzing it. And the only point that I would make is, to do this, we do need the sophisticated computer science that we've talked about here, but domain expertise in biology is critical to marry together with it. One of the reasons, in my view, Watson has really failed to realize its potential is because IBM never recognized the importance of medical domain expertise in a serious way. Thank you very much.
Thank you. So one question, I'll let you go. So this is amazing work you're doing. It's really inspiring. Every time I hear this story, I love it. And I learn a little bit more. So given where you are now and what you've just described, and the dependency upon big data and finding patterns and pattern discoveries in that data, how do you see working with us?
Well, I think the point I should have made that was not clear is we do have an enormous amount of data, but in the fascinating things I've showed you here, we're just barely beginning to scratch the surface in what we can do with it. And I think you raise another possibility, especially for being able to integrate information at different biological levels, to bring us back to a network understanding of the nature of wellness processes and of disease processes. So I think anything new and exciting, that's what we're talking about today in pattern formation, I welcome and will joyfully collaborate with.
Thank you very much. Thanks, Lee.
Okay.